I just had a question on the chat from Gabriella. Thanks. Hang on, let me get the chat back up. It just disappeared. Were these original wild populations very different from each other? And how does that impact the insurance population in terms of retaining local adaptation genes or genetic variations? And that's a fantastic question. And so the original wild populations, in terms of how different they are to each other, it had been assessed for some of the major population or sampling sites on the east coast and the west coast of Tasmania using just a few microsatellite markers, and found that there seemed to be maybe 2 clusters. So they did group separately the eastern populations and the western ones. And a more recent SNP study, which had sampled just a few animals at a few more populations, so around 5 animals or so, found possibly 2 or 3 genetic clusters, but there hadn't been sampling at the time we did this study. There hadn't been more comprehensive sampling of the rest of Tasmania, and most of it had been done with microsatellite markers, and just a couple of them where we may not necessarily pick up fine scale genetic differentiation. So that's really was one of the key questions of this study, was to see, well, what does the genetic variation across the wild range look like in contemporary samples? These were all recent samples as well. In terms of the second part of the question around how it might impact retaining any local adaptation, that's also a really great question. So the study had, in the study, we actually had 2 different data sets. So we had the DART-seq data, which we use to investigate genome-wide diversity that we'll be working with today. We also actually had a functional target capture assay, which looked at sequencing genes that we knew may be of interest. So genes to do with reproduction, immune genes, and also some SNPs that had previously been identified by other groups as potentially being associated with the disease or response to disease. And so that was also a key question, is whether the insurance population captured that diversity as well, and whether there were differences between the populations. So we won't so much be talking about that aspect of the study today. We'll be focusing on the kind of genome-wide diversity and the essential biodiversity variables, but definitely a good point to think about throughout the workshop. So thank you for bringing that up. Did we have any other questions in the room or on the chat? Feel free to ask at any point if you would like to.
No, it seems that we don't have any question for the moment.
Great. So we can move on with the workshop then. So I'll give you a few minutes to set up your R. You hopefully know how to do all of that from the last couple of days of the workshop. I'll share my screen so that we'll work along the code line by line. I should say there's not a whole heap of code in this workshop, so we're going to focus more on understanding the outcomes of the results and why we're looking into this. So it's actually, hopefully, should be fairly straightforward to run the code, but please do let us know if you have any questions as we go. All right, can you see my screen?
Yes,
yep.
Yes, we can see.
Great, thank you. All right, so it's also available in that tutorial format as well, but first we can run the R code. So we'll give people a bit of time to get that set up. So the code uses 2 different, yep, sorry, Ben.
Could you please increase the font size of,
yeah,
the
sure.
RStudio? Please.
Is it this one?
If you go to...
Oh, hang on. Oh, now
If
I've
you go
lost my
to
window.
tools,
To what, sorry?
right, in the menu tools, is in the just before
Yeah.
help. Yes, there. Then global options at the end. Yes. Then appearance in the left. Yes. And then yes, can you put it in 14?
16?
Yes. Yes, and then okay. Yeah, maybe that looks better. Yes, thank you very much.
Thank you. Okay, so yeah, this code relies on two different R packages, so the Dart R first and also the higher FStat package as well. So these should already be loaded into this R environment, but just so you know, these are the packages that are used. We can either run line by line, or you can run the chunk with the play button. And so you'll see that that's loaded the packages. So as I mentioned, this data is a subset of the genome-wide DartSeq data that's publicly available in the study. And as Catherine introduced, the aims are to investigate the current genetic diversity of Tasmanian devils across their entire wild range, and whether the genetic diversity of the insurance population is representative of that wild. And so some first things to do, so I should say this data has already been, so this data was aligned to the Tasmanian devil reference genome. The SNPs were called and have already been filtered for various different quality aspects, which I think are covered in some of the other workshops this week as well. So we're not going to go through the SNP filtering today. But it's still important to investigate what our data looks like. So the first thing are some questions around how many SNPs are there, how many individuals are in the data set, and what is the distribution of individuals in these populations. And so first we'll load the data. And it is a GenLite object, so that is the format that is often used for the different Dart-R functions, as I'm sure you're becoming familiar with. So we can see it's a large GenLite object. And if we just, it's called GL, so that's the name of our object here. If we just run GL, we can see a description of our GenLite object. And so this is always useful to make sure that your data has loaded correctly and is what you are expecting it to be. We can see here it says there are 300 genotypes with 1,663 binary SNPs and 7% missing data. And so we have 300 individual labels here, so 300 individuals in our data set. The study had, I think, over 1,000, so this is a subset, as I said, at 1,663 SNPs. We can also look at the distribution of individuals in populations. So the population metadata is stored in this @pop object of the GenLite. And so if we run the summary of the GenLite, we see that each of the populations here has exactly 20 individuals. So I have subset the data to be that way and selected 20 from each population. Some of the populations only had 20 animals sampled, but some of them, like the insurance population, had closer to 300. So for the purposes of this, we've subset it to 20 each. So we have an even distribution of sample sizes across the data set. So in situations like this, where you have multiple different populations, and I'm assuming you may not be an expert in your Tasmanian geography, as I'm not really either, it can be helpful to allocate colours to the populations for the purpose of graphing and interpreting the data more easily. And so, as Catherine introduced, with the map, we can I guess this is fairly arbitrary. We don't have much pre-existing knowledge around how the genetic diversity differs across Tasmania, other than to know that it seems that there's kind of an eastern cluster and a western cluster at this point, at the time we did the study. And so we can allocate the colours based roughly on the location of the different sites, although this is just without any real prior knowledge of how these sites may be connected or not. And so for this example, we are allocating the colours based on eastern Tasmanian sites, the central sites, and western Tasmania, as well as the captive breeding program. So that encompasses the insurance population and also the Maria Island samples. So first, we can see which populations are included and their order. And so if we run levels, you will see which categories are included in our population, which is the same as what we saw with the summary function, but just doesn't count them up for you. And so I've created an object here called TasCols, where I've allocated 4 different colors. So a blue for the central populations, green for the eastern populations, and yellow for the western, and orange for those captive breeding program populations. So you don't have to remember which ones are which, so you can take my word for this, but it does match up to the map that we showed earlier. And so here I've just done them in the same order as how they were listed in the POP object. So I mentioned that we aren't going to go through the SNP filtering today, as this was done prior to this data set, but it might still be useful to look into patterns within the SNPs at the different populations. And so, for example, we might want to report the minor allele frequency across populations. So I'll give you some time to run this code and have a think about if you notice any patterns in the minor allele frequency, and if there are differences between the overall minor allele frequency distribution, so that's the MAF distribution across the entire data set, compared to that of individual populations. You'll note when you run this function, because it does report both tables and graphs, if you're running it in the window here, you won't output the graphs very well. So you may want to run it in your console. So if you just copy it down to the console and run here, it will send the graphs to the plot window, which you might want to also make a bit bigger so that you can visualize that better, because there are quite a lot of populations to graph. So I'll give you some time to run that, and just, let me know if we're going too fast or if people are wanting to move ahead or so on. So even on this window, because I don't have heaps of room here, it doesn't look very interpretable. It might look a bit nicer on yours. There we go, it's rendering. I think in the tutorial as well, you can see, let's go up to a nicer graph there if you'd like. So if you're finding it hard to visualize on your small laptop screen, you could.
While you've got that code open, I could perhaps share screen with the graph so we can look at it together in a bigger format.
Yep, sure. I will stop sharing.
that look all right?
Yep. Does anyone have any thoughts on the minor allele frequency distribution or patterns that they'd like to share?
It seems pretty clear you've got a lot of very rare alleles, but then after that it just kind of flattens out and it's random as to whether you've got more alleles at 0.2 versus 0.4 versus 0.1 in many of the populations. So it's not a very even curve from low to high frequency.
Yeah, so we do have quite a lot of rare alleles, particularly in some of the populations where, so alleles that are at very low frequency, so that bar towards the 0, the left-hand side of the graphs. The overall distribution, so that top left graph, that shows the MAF distribution across the entire data set, so not split up by all of the different populations. We see that we still have a number of rare alleles, but perhaps not as striking as some of the populations when they've been divided up. And so one thing to note, I guess, is that minor allele frequency is setting a minor allele frequency threshold is a balance between wanting to still retain alleles that may be rare or found, you potentially could be rare and local to a particular population, but also wanting to make sure that we're not including alleles that are so rare that they're possibly just a sequencing error and don't truly exist. And so although we didn't go through the SNP filtering today, the SNPs were filtered on a minor allele frequency of 0.01, I believe, but that was across the entire data set of over 1,000 samples. So actually quite a lot of individuals would have to have that allele for it to be included in the data set. So there is kind of a threshold for the minor allele frequency that also depends on how many individuals you've sampled to set that to balance the sequencing error with possible genuine rare alleles.
So I was just going to throw in a comment. So the other thing you kind of need to know to interpret this is how many alleles are there in each population. So if you're dealing with very small numbers of alleles versus very high number of alleles, you may interpret that table a little bit differently.
Yep. Catherine, do you have a comment on that?
So do you mean whether some of those loci are monomorphic in some populations?
Yeah, so I guess the number of polymorphic loci per population. If it's really low in each of those populations, but the sum of them all is a bit higher in the
Yes.
left-hand graph.
Yes, and a lot of the zero peaks that you're seeing within populations probably accounts for that. There's at the global data set level, there will be SNPs that have a minor allele frequency that's quite low, therefore meaning that if those alleles aren't found in all populations, then those populations will look monomorphic for those. So this graph sort of also shows that there's probably a lot of loci that may be polymorphic in one locality, but not another. So yeah, it's a trade-off. Like Kate was saying, when you've got a data set like this, where the sample size is huge and you've got really broad sampling across the landscape, you want to set your threshold low enough that you can avoid too many sequencing artifacts, but not so low that you end up removing things that might be unique to certain populations. There's a signal to noise issue that we often have to wrestle with the filtering. So there's, oh, go ahead.
I was going to say the question in the chat about would you use a 0.01 MAF threshold if you have only 10 individuals per population. So I should say the MAF filter was applied at the overall data set level, not within each population, which is related to what that previous question was, which is that we will see some populations where they're monomorphic for that SNP because the threshold has been applied at the overall data set level. So that does, by applying it at the data set level, in this case, because we were interested at looking at patterns of genetic diversity overall, we didn't want to apply MAF filters at individual populations, and also some of the other populations in the study or sampling sites had smaller sample sizes than 20 as well. If you start to apply a MAF filter that's too high within those populations, you'll actually knock out your rare alleles or your less frequent alleles, and then it will become difficult to uncover patterns of potentially fine-scale genetic variation because you're now looking at an analysis of just the more frequent alleles. And so, yeah, there was a MAF threshold applied across the entire data set, but not within these individual populations, which is why we do see some in particular where there's very high levels of either monomorphic SNPs within a population or rare alleles. And good observation, Bree, about some of the different populations that their patterns as well. Okay, so Catherine, did you have anything else to add there?
Oh, no, I was just going to quickly comment on Gabriella's question about whether how would you choose the MAF threshold if you only had 10 individuals per population. As just in addition to what Kate said about if you're looking at making comparisons among populations, it can make sense to do your filtering at that global level so that the data is internally consistent so that you can compare among populations within the data set. If you only had a small sample size of 10 or so individuals for the whole study, or perhaps if you were interested just in a within population analysis and weren't necessarily making quantitative comparisons to another population, then the sample size, if it's very small, can be a really important determinant of where you're going to set that filter, because, you know, with only 10 individuals, the rarest an allele could be is 1 in 20, which is 0.05. If it's found in a single heterozygote. So if an allele is found in 2 individuals, then the rarest it can be is 0.1. So you can see that those numbers start to intersect with each other when the sample sizes get very small. So just to add to that. Thank you, Kate.
Yeah, because if there was only one, if we're setting such a low threshold that only one individual has that allele, then we can't really trust that it is a true allele across the population and isn't just a sequencing error or SNP calling error. So the threshold does depend somewhat on sample size as well. Great, so I'll go back to sharing the code. So one of the first things that we want to do is to visualize population structure. And in this case, we'll be doing that by PCOA. And you have another workshop later in the week, I think, on different distance metrics, which will go into the theory behind PCOA and some of the other measures in a lot more detail. And so PCOA, we can do this to describe patterns in population structure, but it is a visualization. And so some questions are, how would you describe any patterns in population structure, and how does the population structure of the insurance population in orange compare to that of the wild, remembering that that was one of the key aims of the study. And so we first need to generate the PCOA. So using this gl.pcoa function with our genlight object, we can run that, which is performing the PCA. And then we can plot the PCOA once that's finished running. And so this is the default data visualization for PCA. And so we have, if you're not so familiar with PCA so far, we have axis 1, which in this case describes 10.1% of the variation in our data. And we have axis 2 describing 3.2% of the variation in our data. And you can see that it assigns the labels to the different populations and automatically colors them with the default shadings, which is very helpful, but it can be a little bit tricky to figure out where those, where the labels correspond to the points. And also at the moment looks a little bit difficult to interpret compared to our thoughts around the geography of Tasmania and the sampling locations. And so using those colors that we set earlier on, we can actually visualize the same data, but using the color labels that we supplied earlier. So that object that we made of the Tas colors, we can rerun the plot with assigning the point colors with that object. And so we can see, well, I'll leave it to you to see what you think about the patterns. And so we have the populations. I'll just remind you of which geographically, which ones these were. So the green represented the eastern populations. The blue represented those central Tasmanian populations. Yellow was the west coast populations. And then orange is the insurance population and Maria Island. So going back to the questions then, how would you describe any patterns in population structure? And how does the population structure of the insurance population compare to that of the wild? So I'll give you some time to run that. And then if anyone would like to comment or put questions in the chat as well.
Hi, Catherine. Thanks for this. My name's Amy. I just wanted to ask, or maybe make a comment that sometimes when I've run PCOAs, if you remove like one of the clusters, you might find further structure or further subsets within those clusters. Is that something that you would look at as well?
Yeah, definitely. So here we're visualizing the overall structure, I guess, across our data set. So we haven't, so it's describing the overall variation in the data set here. Firstly, this is just PC1 verse 2. So I guess one thing is that you can also then look at PC2 and 3 and so on. So you may want to look at additional principal components to just 1 and 2 that describe the most variation in the data set. I guess the other thing to note is that if we were to remove, for example, all of the populations, like all of the Western populations, it would then just be describing the variation in the remaining data set, and you may get quite different patterns or a different visualization because of what variation it's describing now. So this really is only looking at the variation in the entire data set. And if populations, for example, if you had a population that looked very different to all of the other populations, it might kind of cluster together very tightly compared to the others and away from them perhaps. But if you then were to make a PCOA of just that population, there could be population structure within that population, if that makes sense. So it is impacted by what's in the data set because it's describing the overall variation. Hopefully that answers your question. Thanks, Amy. So any comments on population structure or the insurance population in comparison?
Would you like me to put the map back up for a moment?
Yeah, that might help. So you should hopefully have the PCA on your own screen. So you compare that to what Catherine will show.
Maybe even show both.
Maybe there's some gene flow between is it the north and the south, the green and the blue? Oh,
Yeah,
sorry,
so green we've got as the eastern
and the central.
eastern population. The yellow is the western population, and blue being that central, sorry, not population, locations. And so I think we can kind of see that the colors, so when we shaded that first PCOA we did where everything was just a default color, it's a bit hard to tell, I guess. There is some population structure perhaps, but it's a bit hard to tell how that might be influenced by geography, if it is at all. And so we've got some great thoughts coming through in the chat, so we'll get to those as well. But what we can see when we shade it by the geographical assigned colors that we've used, we do see that it is, we can see those green individuals seem to kind of be grouping roughly together. The blue ones, the blue dots are all near each other. They're not scattered all over the PCOA, and same with the yellow. So it does seem that there is genetic structuring across those different regions that we've assigned here. It's not just like a mess of different colors all over the PCOA. So I'll just go through some of the comments in the chat as well. So someone said the insurance population seems spread across all three, so I assume all three other groups, but perhaps closer to the Western population. So we can see that there's quite a lot of orange dots overlapping those yellow ones there. I think that's a good observation. So we do see the insurance population does appear to represent those different genetic groups by PCOA, but we do have a lot of clustering around that, the yellow western side. And that also reflects the sampling of animals that came into the insurance population, where they were mostly taken from the west of Tasmania ahead of that disease front until more recent years, where orphaned animals that didn't have DFTD were brought into the breeding program from the eastern populations as well. So it does seem, Dylan said it seems like a decent spread, perhaps a bit biased towards the yellow. So yes, it's good observation. We've got a question about how many generations of breeding had occurred within the insurance population at this point. So it began in 2006, the insurance population, and devils have a generation time of about 2.5 years, so or 2 years. So the sampling was done mostly around 2018, 19, 20, I think. So that's a number of generations of captive breeding at that point, although it has been genetically monitored throughout. We've got comments around how the eastern and central locations seem more continuous with some intermixing. So that may be partly to do with how we've assigned those populations or those groupings. By, it's a little bit arbitrary where we draw the line about what is eastern and what is central.
Hey, could I jump in there?
Yeah,
I think that's a really good point to talk about how we identify our population identities, or how we design a project like this, where we might have a priori reasons for defining our hypothesis about the populations that we have. And we might make those hypotheses on the basis of a variety of information. In this case, it's a combination of previous data. So as we've mentioned, there was microsatellite data from the East and West that suggested there were at least these two Eastern and Western clusters, as well as some other targeted sampling projects. We might make that determination based on ecological data. So knowing what's known about the landscape and how devils move around in the landscape, what is a plausible barrier to movement, and so on. So it's known that devils will use roads, for example, to traverse the landscape. So that might help to make hypotheses about which localities are likely to be separate from each other. And then as well as strategic priorities. So from the purpose of the devil program, like Kate said, working with the Citizen Science Project to strategically and deliberately get samples from places that hadn't been previously sampled, as well as in a sort of practical sense, just sort of saying, well, look, these populations, knowing all of those other factors, these populations are reasonably close together. So let's sort of come up with an initial hypothesis that they could well be connected. So for the purpose of the workshop, as Kate has already said, this is a subset of the data, and we've tried to keep it simple and just sort of say, look, there's a Western, a Central, and an Eastern, but you can see how with your own study system, or even with this one, it would be possible to be a lot more refined about how you set those hypotheses up. And certainly if you wanted to learn more, there's plenty of other devil papers that we can share with you that talk a lot more about how devils move in the landscape. But yeah, when you're thinking about how you're going to set up your localities for your own studies or for your own other systems, I would remind everyone to draw on a variety of data sources to come up with those hypotheses. And I think later we can also, later in the workshop, you'll also see some methods for sort of generating hypotheses about population structure, where you don't necessarily define a priori what all your localities are, but instead use the genetic data to tell you where the genetic clusters are and whether or not different samples likely represent different populations.
Hi, there's just a question in the room. My name's Matt. I just wondered if you may have get a better result with your PCA if you add 3 dimensions to it?
In the one graph, or to look at
PCA
PC?
plot, yeah, just what happens in terms of the way it appears to overlap with that insurance population. Is it actually really overlapping, or is it far off in the distance if you plot it on a third dimension?
Yeah, great question. So there is a function to do the 3D plots, I believe, as well. I personally find them just difficult to visualize in a 3D plot, but I would usually look at the axis 3 as well, not just 1 and 2, depending on how much variation. I don't know if someone's going to go into the distance. I think perhaps Arthur will in his workshop, the distance metrics and how the eigenvalues and so on work for PCA. But that's true, that just because it's overlapping in this axis doesn't mean that it truly is overlapping and that it isn't genetically differentiated in a different way. Might relate to Bree's question as well around like the percentage variation described. So in axis 1, we've got 10%, in axis 2, 3%. They're not huge values for the amount of variation described. And Hannah, I think also like great interpretation about PC1 splitting the Western compared to the Eastern and Central populations. It seems that is what's driving the variation in PC1, whereas PC2 is perhaps differentiating the Eastern populations from the Central ones. But yeah, without looking at PC3, we don't know whether there's another layer of variation there. Does that answer the question, Matt?
Cheers. Yes, it does.
Yep.
Thank you.
Thanks. And in terms of the amount of variation that's acceptable, I do see quite different amounts depending on the data set, the species and the populations you're looking at. So it's not really that there's a rule around what is acceptable. Quite often see values higher than this, but not necessarily as high as 70% that you might see with some environmental variables. I have seen that before, but that then gets to the question, I guess, of whether things are even different species, perhaps. But there can be reasonable explanations for that. So particularly if there's a couple of very isolated populations that have drifted, for example. So there is a lot that can drive the variation within that we see in a PCA. And it's certainly not the only metric that we should use to assess populations and structure. But Tasmanian devils are kind of notorious for having very low genetic diversity. And so although these are quite low percentage variation, it actually was somewhat surprising to us how much fine scale structure there was in this data set. So for the species, but perhaps compared to other species is not very much. So thanks everyone for your input on the PCAs. We can perhaps move on to the next part of the code. So if I share that again. So yep, we were going to look at one of the essential biodiversity variable metrics that Catherine introduced. So she talked about how genetic richness is one of the components for genetic monitoring. And so we can calculate allelic richness for each population. And allelic richness, you may, if you've worked with microsatellites before, you have probably used this metric because it's very useful, particularly in microsatellites or if you're looking at particular genes. It's not as commonly used with SNP data, as you'll see why, but we can still run that analysis. So it requires the higher FStat package. So there's not a function in Dart-R at this point to do that, but it's a very easy conversion. So we first need to convert the GenLight Dart-R object that we have to a GenInd format with the GL to GI function. So that gives us a GenInd object. If we run that object, you'll see you get a similar summary to when you run the GL summary. So just a summary is different format of storing the data. We then need to convert it to the right format for the higher FStat package. And then from that, we can use this allelic richness function of the higher FStat package to calculate allelic richness. And so we've run that and called it AR, our object. If you use the names function, you can see what attributes the object has. And it has something called min all. So if we run that, we get 12. So what that refers to is the rarefication of the allelic richness calculation, which is based on the smallest sample size for a genotyped locus. So this will vary depending on the polymorphism in your data set. But that's what the calculation of allelic richness has been rarefied to. If we then run a summary of the AR part of the object, the allelic richness, we get the basic summary statistics for each population. And we could look at the mean, for example, to compare the different populations. So one thing you'll note is that for all of the populations, the minimum is 1 and the maximum is 2. So that is the case for this SNP data, where there's either 1 or 2 alleles. If you were using microsatellite or other types of data, that would be more variable. And so we can then, perhaps if you just wanted to pull out the means rather than the summary of everything, we could do that with the column means, and we'll get an object or a value, sorry, of the mean allelic richness for each population. As an example here, I've given some code for how we would also calculate a measure of variance. So if we're reporting statistics such as allelic richness or any of the statistics, it's also really helpful to report a standard error or measure of variance of some kind. So either a standard deviation or standard error. And here the standard error we can calculate as the standard deviation divided by the square root of the sample size, where the sample size is the number of genotype loci. And so I've done this for Bronte, one of the populations here, but you could make that into a function and loop it through all of the populations, or you could just copy and run it for different populations, but it's more just there as an example. This isn't a very helpful way to visualize our results, so I prefer to visualize. So we could make a box plot of allelic richness per population and have a think about which populations have the highest and the lowest allelic richness. And if you can think of any explanation, possible explanation for why. With this graph, the first thing that we're doing is actually setting the margins of the graphing window. So if you're not familiar with this, the margins describe like the space from the bottom and then the sides and the top of the graphing window. And here we've just extended the bottom margin to 8 because I've got these long population label names. And if we don't extend it, they'll just cut off and you can't read the full. We could make them smaller, but if we want them to stay large and don't want them to get cut off, we can extend the graphing window and run this box plot. And so you can see it's not a very useful visualization, a box plot, when it goes from 1 to 2. But we do still see the trends, I guess, in allelic richness between the populations. Given that we've color-coded them nicely, it might be nicer, I think, to visualize them grouped together. So if we group the Eastern, Western, Central, so we can actually reorder our allelic richness measures by the order of our populations, which I've manually coded here. And we can reorder the colors so that the colors are assigned to the correct population, and then rerun that box plot. Let me also run this. So you can see we get the labels as well. So now we've actually grouped the Western populations, the Central, the Eastern, and the insurance population in Maria Island together. So any comments on the trends there?
Not many.
Yeah. So I guess one thing we were particularly interested in was how well the insurance population was doing compared to the rest of the wild locations. So we do have a reasonably high allelic richness in the insurance population, which is reassuring there. Someone's commented in the chat, Bree, thank you, that blue has higher allelic richness compared to the west and the east, so that's essential. We do see, so some sites in particular seem to have very low allelic richness. So Robbins Island and Woolnorth and the Tasman Peninsula in particular have quite low allelic richness. And what you might be able to pick up from the names of those locations is, so one is a very small island. The other is a peninsula that's quite cut off from, there's not much movement of Tasmanian devils through the peninsula. And so that may indicate why those sites in particular seem to have quite low allelic richness. Someone's asked, was Lolita, was site location designated in the individual metrics file or in the code at the start of this analysis? So the population was assigned in the GenLite object. So that's the GenLite at pop will give the population. So whether it's Robbins Island or Woolnorth or Granville Harbour or so on, the location that we've grouped them into, we assigned with the code later. So it's not actually in the we could update the population metadata to have that instead, but we're still interested in the population level comparison. So I've left it like that for now. But good question. All right, if there's no further questions on that, we can move on to calculating heterozygosity statistics for each population. And so there's, I think, a function that you may have already used in the workshop or will definitely come to use at other points as well, GL report heterozygosity. So this reports the observed, expected, and unbiased heterozygosities and inbreeding coefficient measured as FIS, either by population, or you can calculate the observed heterozygosity for each individual in a GenLite object. And so we can calculate these statistics for each population, which is what we were interested in. So this study was a population level analysis. We weren't really looking at individuals. And then some questions are, which populations have the highest and the lowest heterozygosity? And within a population, do you notice any patterns of observed compared to expected heterozygosity from the graph? And what about FIS, so inbreeding? And with these functions, you can also use the question mark to run to get the help information. So whenever you go to run a function, you can just use that help or you can search for it in the help bar here. And you'll get the documentation about what are the different options that you can supply to the function and what each of those options means, as well as the details of how, this is a really well documented one, thanks Dart people, the details of how these metrics have been calculated in this case, the output, and also often some, oh, this one's just so good, tutorials. Also often you will get examples of usage. So if you're not sure how to run a function, at the bottom of the help function, you'll get some examples. So that's just something to note when you're using R if you're new to it as well. So having read this very helpful documentation, we know that we have a few different options. We need to supply the GenLite object. We need to tell it which method to use, or by default, it will use population, which is actually what we want to use anyway, so we don't have to specify it in this case. And we also want it to plot the colors of the populations using the vector that we've already created so that we don't just get the default colors. So we can run that function. And it outputs a number of different things. So you actually do get the data frame, the table of what the values are for each of these statistics. So we can see we've got the values here, and this is our object hets. So we could also open it if we wanted to look at these values, or we could export that if we wanted to save that as a CSV, for example, and have all of the values. We also then get the visualization, so the graph of observed heterozygosity, expected, and FIS. And this is just in default order, so ordered. I think it used to be ordered a different way, but maybe it's the version of Dart R. So if we go back to the questions about which populations have highest and lowest heterozygosity, and whether you noticed any patterns within a population of observed compared to expected. So we can see visually the one with the lowest is Robbins Island, that island population on the northwest Tasmania. Amy's made a good observation that the western populations on average seem to have lower heterozygosity than the eastern and central. In terms of the ones that have the highest heterozygosities, we can see Buckland, one of the eastern populations, quite high. and so is the insurance population, which is reassuring because obviously we want to make sure that they're genetically diverse. It can be a bit hard to compare the observed and expected heterozygosity visually from this graph when they're not next to each other, but if we perhaps were to look at the table instead, actually maybe we'll just look at it here. We can see, sorry if those numbers are a bit small, but maybe you can look at your own screen as well. We can see that some of the observed heterozygosities do seem to be lower than some of the expected heterozygosities. And in cases where they are, that is then where we see these positive FIS values. And that's to do with the way that inbreeding is calculated in this function, where it's looking at observed and expected heterozygosity. So just to demonstrate that we can also use the same function, but switch the method from POP, so to calculate these statistics per population, to IND, to calculate it at the individual level observed heterozygosity. And this can be quite useful if you're trying to investigate individual level diversity, which you might be, for example, if you're looking at a very managed population where you know all the individuals, like a captive breeding program. But it can also be useful in assessing the impact of sample quality and missing data on your results. And so we can run the same, so exact same function, but using this method individual to get individual heterozygosities. And once again, you'll see you get some information about running the function. We also get the table, which if we were to view that, we have for every individual in the data set, the observed heterozygosity, as well as the number of loci that it was genotyped at. And we get a graph that shows the distribution of observed heterozygosities across the individuals. So we can see there are some outliers, in particular, a few outliers that have very low observed heterozygosities compared to the rest of the data set. Does anyone have any thoughts on what some explanations might be for that? Feel free to put them in the chat or speak out if you would like. So, Julia, on the
We're
chat.
making this as difficult as possible here. I just said shit DNA is part of the reason you get bad call rates on some individuals.
Yeah, you've put it perfectly. Yeah, so often poor DNA quality going into your sequencing of samples can lead to low heterozygosity at that individual sample. So actually, this is something that really should be assessed early on in your SNP filtering, also looking at individual samples to make sure that if you include the sample in your analysis, it's not going to be dragging down your population level estimates, for example, just because of a couple of samples that could be not very well sequenced. Christine said it could be indicative of inbreeding, so that's also true. There are genetic explanations for why there could be low levels of heterozygosity. So I think one thing that's useful is to then look at, so exactly what Catherine's just said in the chat. So other metadata you might have for that sample, like going back to your lab quality control, the sequencing metadata, and also the amount of missing data. So we can actually look at the number of loci. So straight away we can see here, for example, this individual from Maria Island has a very low observed heterozygosity, so one of our outliers, and also has the lowest number of loci that it was genotyped at. So that probably is the explanation for that sample rather than it being a genetic indication. So definitely, we've gone to sleep. Sorry, but definitely something to consider. So you can move on with the next chunk of code while I reload this. Hopefully it's saved my objects. Oh, it has. Excellent. So Gabrielle has asked, if missing data is low, how would you identify if it is cross-contaminated? Cross-contaminated as in with other sample, like another sample in sequencing, or?
One thing you can do is, if you know the sex of the individual, you can look for perhaps any SNPs on the sex chromosomes that might help you identify whether it might be sample mix-up.
Oh, for an individual with high heterozygosity. So we do also, so DartSeq also sequences technical replicates, where they sequence like a sample that you've sent, they'll sequence in duplicate. So the exact same DNA that you've sent them in the well, they will sequence twice. And therefore, we've got a good technical comparison for what the genotypes should be between those two samples, which is what contributes to that rep average column or the reproducibility column in the DartSeq metadata files or the spreadsheets you get back, and is one of the filters that we often filter SNPs on. So that helps, I guess, to make sure that you're seeing expected similar, like the same genotypes between technical replicates, and removing any loci where replicates that you would expect to be the same consistently have different genotypes. So I guess that's one way to reduce that possibility, is to filter on reproducibility to make sure that you're getting consistent genotype calls across your replicate samples. Or if you're doing a different type of sequencing, make sure that you send replicates as well so that you can make that comparison. And then so final question in the workshop for the practical part is around FST, pairwise FST values. So these are a quantitative measure of genetic differentiation, which also have their flaws, or has its flaws, but we can calculate this pairwise between the different populations to see which have the most genetic differentiation according to FST, and also have a think about where these populations are located. So we can run this with 4 cores if your machine has that available to make it speed up a little bit. And then we're using this gl.fst.pop function with 100 bootstraps. So you can increase this to 1,000 is what I would typically run, but we'll do 100 so it runs a little bit more quickly, and 95% confidence intervals. And then we can make this into a nice table with 3 decimal places. So we'll run that there. And then it might take some time in the results for you to figure out which populations have the highest or the largest pairwise FST values, because there is quite a lot of populations to compare. So we can see our table is a matrix of the populations on in rows compared to each population going across the columns. So obviously it's NA for a population against itself, but then we have the FST value between, in this case, Maria Island and the Forestier populations. And if we scroll along, you can see that this continues across the data set to compare each of the different populations pairwise. So while you're looking at that to see which ones have the highest, I might ask Catherine to please pull the map back up. I'll stop sharing my screen. And once you have figured it out, maybe drop that into the chat or let us know what you think.
And I think we've lost connection with the Zoom room, but the online people are still here.
Oh no.
I'll just see if I can troubleshoot that.
I'll be back.
Hello, Catherine. Kate, can
Yep.
you hear us?
Yes.
Oh, we're back. Great.
I'm sorry. Yeah, we have this hiccup, but I think we are almost done, right?
Yeah, almost. This
We
is just the last
Yes.
question. Yeah. So I'm not sure when we dropped off. So the last question was just around which populations have the highest pairwise FST values. So that was that last chunk of code. And a couple of people have said in the chat, there's a few with very high FST values between them. So Robbins Island and Forestier, and then also Tasman Peninsula and Robbins Island. And if we go back to our map and look at where those locations are, we can see so Robbins Island up in that very northwest corner, and an island, so isolated population, and Tasman Peninsula in the very kind of southeast of our sampling range. That's also fairly isolated along that peninsula there. So we do have a reasonable explanation for why those populations would be so differentiated, but also probably the effects of drift perhaps, or inbreeding on those islands that may also contribute to high pairwise genetic differentiation values. So that's it for our formal part of the workshop and the questions. So Catherine, did you want to go back to the objectives if you've got those? And so I think we've shown, in today's activity, we've shown you how to calculate some of the essential biodiversity variables for a subset of the populations in the study we've used, and to consider the implications of the sampling and study design for addressing these kinds of questions. In terms of Tasmanian devil management, this really was a key study for answering the questions around the value of the insurance population and genetic diversity there, but also identifying what the genetic landscape looks like across the species at a finer scale than what we had previously done, or what other people had previously looked at with microsatellite markers. And if there's time for any questions, we're happy to take questions, but thanks very much, and thank you to Bern and Lewis for organising such a great workshop as well, and having us speak. Thank you.